Now, the nitrogenous bases themselves, I've already distinguished as generally being called a pyrimidine version or a purine version. These are heteroaromatic molecules. That is key. They are aromatic, and that is because of these alternating double single bonds that exist within the ring. They're pretty much planar. They're pretty flat and they're aromatic. And because of this, they absorb UV light at around 250 to 270 nanometers. Excuse me. Why is this important? This is important because it lets us track DNA in a sample. I have brought up absorbance numerous times. This is when talking about proteins and now also talking about DNA. And the reason that it is brought up, the reason we talk about where things absorb UV light is because when we as scientists are measuring how much DNA is in a sample or how much protein is in the sample, we need something to be able to measure. And one of the main things that is utilized as a source of quantitative data is using UV spectroscopy to measure how much of a biomolecule is present. We talked about in proteins, proteins also have aromatic amino acids that in that case happen to have a UV absorbance of around 280. This is good because it helps us distinguish if we have protein or if we have some type of nucleotide. Nucleotides, again, are going to absorb around 250 to 270. So if you have a sample, you can look at your 250 to 270 range and decide how much DNA and RNA and nucleotides you have in a sample. And then you can look at your 280 absorbance and decide how much amino acids or protein you have in your sample. This is very, very, very common technique. Now, the actual bases themselves are categorized based on small differences between their forms. Within the pyrimidine bases and the purine bases, we have a total of 5 commonly seen bases. There are cytosine, adenine, and guanine, which are found in DNA and RNA. Then there's also thymine, which is only found in DNA, and uracil, which is only found in DNA. So essentially, these two are swapped for each other depending on if you have DNA or RNA. The nitrogenous bases themselves are all very good hydrogen bond donors and acceptors. And this is important because the way that our polymers are going to be formed in our nucleic acids are based on these hydrogen bonds. And they're all neutral molecules at a pH of 7. So they do not have a charge on them. That's just the nitrogenous bases themselves, not the phosphate group. And if you remember, we said the phosphate group actually does have a charge on it, has a -2 charge on it in a pH of 7. So the actually distinguishable groups that we have are in our purines, adenine and guanine, which is A and G respectively. You can see the difference between adenine and guanine lays mainly in our groups here and here. So you can see adenine has an amine group on its top carbon and no group, just a hydrogen here on this first carbon here. Whereas in guanine, we have an amine group And we have up top a carbonyl group. Otherwise, their structures are fairly similar. Now, where do these rings attach to the sugar? On adenine and guanine, they utilize this nitrogen, usually drawn on the bottom left nitrogen, and it's drawn there in particular in that position normally as a way to remember that this is where the nitrogenous base attaches to the sugar. Now for the pyrimidines, we have cytosine, thymine, of course, if you're just in DNA, and uracil if you're just in RNA. Similarly, these groups are distinguishable mainly by those same carbons in these positions, mainly. You can see that across the board, they all have a carbonyl group on this carbon. Cytosine has an amine here. Thymine has a carbonyl, and uracil also has a carbonyl. So you'll see now that thymine and uracil, from what I have circled, look identical. And the only distinguishing factor between thymine and uracil is actually this methyl group here. So this methyl group is what distinguishes thymine in DNA from uracil in RNA. And those are the main, all of the main nitrogenous bases. And memorize these structures, the ones we just went over. So adenine and guanine, the purines, and cytosine, thymine, and uracil, the pyrimidines. If you memorize these nitrogenous bases, based on what we've gone over previously, you should be able to put together a whole nucleotide. We know what sugars look like, and we know that we use ribose or deoxyribose. And now we know what our nitrogenous bases look like and where our phosphate group attaches.